everyone. I'm Kelly Bachovchin. Thank you for the introduction. Um, as Marcus said, I have been working with CDD Vault for a little over a year now. Before that, I was actually a customer of CDD Vault when I was doing my PhD work on neglected tropical diseases. So all throughout my career, actually, I should say, actually in school, I started as an organic chemist, doing my undergraduate and master's thesis in organic synthesis. So the biotech degree is a little misleading because all of the research I've ever done is organic synthesis at the bench. And so, yeah, I'm pretty much today going to be showing you part of my PhD dissertation work on neglected disease drug discovery, which I did with the Palastri lab at Northeastern University. And this was done in collaboration with GlaxoSmithKline at Tres Cantos. So today I just want to sort of summarize how my presentation is going to go. First, I'd like to give you an introduction and an overview, sort of high-level view of CDD Vault. Then I'll give a background, just a little bit on neglected diseases and why we, are working on these types of diseases in these larger, globally spread collaborations. And then I'm actually going to log into the CDD Vault software to show you the platform using the neglected disease research that we did. Okay, so what is CDD Vault? Well, it is an informatics solution. It is a software that is cloud-based. So it is ready to use in 90 minutes or less. We can spin up an instance. I can do this in pretty much 10 minutes. And then from there, the customers will configure and customize their vault or their container for their data to fit whatever needs that they have. So the vault is suitable for all types of chemical or biological data. As it says here, it can be a combination of both, or really you can, kind of configure the system to capture any type of data. Since it is cloud-based, we have the servers and we fully maintain those servers. So you do not need IT personnel to maintain this software or add protocols and register compounds. It's all done by you as the user. And the platform comes with no additional fees for support and training because we want to make sure that you are getting the most out of the software. So that is actually my role at CDD Vault. I'm on the support team, so I do technical support and I help users basically configure their vaults and train them in how to use the system effectively. So the platform looks like this. It is the software has 4 different modules that work in concert to make up the entire platform. So we have a full activity and registration database capabilities where you can register various different types of entities. This works in hand with our inventory module that helps you keep track of either your chemical compounds that you're registering or maybe a biological entity that you're registering. We also have a visualization module to allow you to look at trends and in your data and do different types of SAR analyses, as well as an electronic laboratory notebook, which is a separate piece of CDD Vault, but it is fully integrated and works very well with the activity and registration platform. So I'm actually going to get through all of these modules when I get to the system itself. But just to show you here the various different types of data you can store within the vault, you can register small molecules with chemical structures, without their chemical structures. this is done using the ChemAxon cartridge. So they are fully rendered structures within the platform. But you can register entities or, biological specimens that are not small molecule compounds and track data associated with those entities that you're registering. So, some examples, maybe you have cell lines that you want to keep track of, or plant specimens, plasmids that you're constructing, antibodies, and so on. And then all of the data associated with those registered entities can also be stored within the vault. Now, within your vault, so that's the highest level container, you can store your entities either in the same vault or in separate vaults, because each vault, again, can be configured and customized to store data specifically for the type of entity that you're registering. And then within each instance of a vault, you can separate your data into projects, which you can label however you'd like. And projects are the way that we allow certain users to see certain data. So you'll see here this, chemist on the right-hand side with the green arrow only has access to the data in project 3, whereas these other 2 scientists have access to multiple projects. And basically, if a person doesn't have access to a project, they don't have the ability to see the data in that project. So CDD Vault is great for working with collaborators that are maybe on 2 out of the 5 projects that you're working on in your lab. Lots of customers also add their CROs into their vault. So, if your CRO is doing your chemical synthesis, they have their own project and they can add data directly into the vault. And again, since it's cloud-based, you can access the database really from anywhere in the world, as long as you have a working internet connection and a web browser, as well as access with a username and a password. So just to summarize the Vault platform, we like to think as it's helping to be complexity simplified. The platform is very intuitive, as you'll see coming up. Like I said, less than 90 minutes to set up the platform. It's a single platform for all different types of data, so you can have chemistry and biological data in the same place. And you have this convenient access through web browsers, allowing your collaborators and all of your team members, especially now during the pandemic when we're working from home, you can access your data, from anywhere. The platform is secure. There is no IT overhead. We fully maintain the servers that the data is stored on, and you allow access to data based on user roles as well as projects. And then finally, we'd like to think of the platform as being collaborative. We do have an application programming interface, so you can connect with other types of softwares, things like KNIME and Pipeline Pilot, to actually push and pull data directly from the system, as well as connecting to other visualization tools, things like Data Warrior and Optibrium Stardrop. It's easy to scale up. We can add vaults, we can add members, or you would actually add members into the vault as needed. And it's very easy to share data that is organized and up to date with collaborators as well as potential investors. Now, just to switch gears a little bit, I want to give a little background on neglected tropical diseases and how we at the Pilastri Lab actually use CDD Vault in our work on developing drugs for these neglected tropical diseases. So basically, neglected tropical diseases, they're diseases that generally afflict the world's poorest populations. And these effects, while the diseases affect people's physical health, they also affect their mental health, as well as create social stigmas, as well as further economic burden on these populations of people who are afflicted with these diseases. So while you may be infected with one of these diseases over multiple years, it definitely affects, long-term the populations in these areas. And basically, research for these treatments is not typically pursued by larger pharmaceutical companies because of the low return on investment. Again, it's affecting the world's poorest population, so you're not going to have, people buying these medications. So a lot of research that's done in the neglected disease area is done through these large global collaborations. We worked a lot with DNDi. I know the Bill and Melinda Gates Foundation does a lot of work on malaria as well as tuberculosis. So again, these are large collaborative efforts, within institutions across the globe. So CDD Vault, being this cloud-based platform, is sort of, the best solution, as well as cost-effective in keeping track of up-to-date data. Now, the disease I'm going to be focusing on today that my work was done on is African sleeping sickness. It's otherwise known as human African trypanosomiasis. It's caused by the infection of a protozoan parasite called Trypanosoma brucei, and there are two subspecies of this parasite, Gambiense and Rhodesiense, and the different subspecies cause either a long-term version that can last over years of a person's life versus a more acute infection where a person may die within a couple of months. So basically, the parasite will get into someone's bloodstream through the bite of a fly that's carrying the parasite. And then eventually, the parasite will cross the blood-brain barrier, get into the brain, which eventually leads to death. So symptoms, are not unlike flu-like symptoms. Sometimes it's very hard to diagnose because lots of people think, they just have a common cold or, the flu, as well as getting to treatment facilities may not always be the easiest. As the disease progresses, as those parasites cross into the brain, you get symptoms more related to disrupted sleep patterns where people are, sleeping all day but awake all night. Eventually, it leads to coma and death. And this disease is 100% fatal if it's left untreated. But the treatments that are available are highly insufficient. They require long periods of dosing. You can have 10 days of IV infusions, which are not easily administered in sub-Saharan Africa, where this disease is prevalent. And most of the treatments cause, really bad side effects. Melarsoprol, one of the only treatments for the the brain stage infection of the disease, is actually 5% fatal in the patients that it's used to treat. It's actually an arsenic-based drug. So based on all of this information, the goal is to find a treatment for this disease that is oral, that is fast acting, kills parasites, so you're not just prolonging the growth inhibition of the parasites, you're actually killing the parasites with this oral dose, as well as being cost effective. So, we want to have, cheaper treatments so that we can distribute the treatments to the population. So that's a background on neglected diseases and African sleeping sickness. And I'm actually going to switch over to my CDD Vault to tell you a little more background on the actual study that was done in collaboration with GlaxoSmithKline, Tres Cantos, and the Plashru Lab at Northeastern. So I'm here logged into my CDD Vault. I'm actually in my ELN. So when you create an ELN page, it's basically a blank page where you can capture all different types of structured or unstructured data. So here I've just used it to summarize this reference article where this assay is described of a high throughput screen that was done at GSK of a kinase targeted library. So kinases, very well known as being druggable targets. So parasites also, or the Trypanosoma brucei parasites also express essential kinases. And so basically a kinase targeted, so a kinase-like list of compounds was screened against whole cell Trypanosoma brucei to see whether or not it inhibited the growth of those parasites. That initial screen is actually summarized here in this assay annotation that I've added into my ELN entry. So this is added in using this little Eppendorf tube. And this assay annotation is actually a mini version of one of our other products, which is BioHarmony Annotator. And it uses a list of thousands of curated ontologies from publicly available biological assays. And so you fill in the different categories for the assay that you're running to, annotate that assay and capture information about how that assay was run, maybe the method, the detector that was used. And then based on the categories that are filled in for your assay, it'll generate a summary at the top, basically to make sure that the descriptions of your assays are less ambiguous. And you can copy this annotation and use it in a publication or in the setup of protocols, which I'll talk about in a couple of minutes. So after the first initial screen of compounds against the whole cell Trypanosoma brucei, they were tested in a dose response assay to find some hit molecules. And through different iterations, the compound series was narrowed down to clusters of compounds based on structural similarity and grouped into clusters and singletons. So that data, I've attached some graphics from that PDF article. So you can attach documents directly to your ELN entries. So I have a PDF article here that I can, expand. If I click on this, I would be able to download it to my desktop and view the full document. Here I have some images from that article that show, how those compounds were narrowed down based on their properties as well as potencies. And now I've actually attached the raw data or the actual data collected from these studies in Excel files here. So we do have an integration with Microsoft Office. This allows you to expand the document here in line with the ELN page so you can see the full document. So these are the compounds and the associated data with the compounds that I have actually already registered into my activity and registration database. So the great part about the ELN and the activity and registration database is that, I can import this data into my database right here from my ELN. Before I do that, I have to set up a container for that data to go into within the database. So I do that through what we call protocols. So now on this Explore Data tab is where the activity and registration platform lives, right? So we were just in the ELN. Now we're in activity and registration. So it's just another tab. And in order to capture your biological assay data or whatever type of experimental data in a more structured format, you'll associate that data to the compounds or the entities that you register in your database. And you store that data in data containers that we call protocols. So different than what most biologists think of when they hear the word protocol, but just understand it as being a container for your data. So these protocols are set up by the users, to capture really all different types of containers. So here is where I would go to set up my protocol. I give it a name and a category and a description to capture the data. And then I would just create the readouts for my data to be uploaded to. So if I just go back quickly to this entry, you'll see I have a column for each of those readouts that I've created in my protocol for this data to then be uploaded to. There are 3 different types of readouts that can be created in the platform. These are all just standard data in, data out type of readouts. So whatever you upload, that's what's going to be displayed back to you when you search for the data later. You can also add in calculated readout definitions. So let's say I wanted to look at the selectivity ratio of my parasite inhibition versus the control cells or the HepG2 cells. So this is not going to have a unit. Basically, I can type a formula into this box and perform a calculation here within the platform using the readouts that I've already created. So if I type an open square bracket here, I can pull up my T-Brucei PEC50 values, and I can divide that by the, HepG2 control cell line and add this calculated readout. And now it's going to use the data that will eventually be uploaded to these 2 readouts to calculate my selectivity ratio for me. The last type of readout that you can create within a protocol is a dose response calculation. And this will actually use raw data and concentrations that you upload to generate live dose response plots. I don't have any of that raw data because I was only provided with PEC50 values. So that's why I don't have a dose response plot calculated, but it is definitely possible. We use the Levenberg-Marquardt algorithm to generate a 4-parameter curve fit when you upload your data. And this can be fully customized. You can also calculate an IC50 as well as other types of calculations based on that raw data that is imported. Once the protocol is set up, then we can import data into our activity and registration database. So I'm going to click Done Editing. And this data now that I've attached to my ELN entry, I have the option with this little arrow here to import this file. By clicking that arrow, it's going to take me to this 3rd tab, which is our Import Data Wizard. So it's a 3-step wizard, and it's used for adding protocol data as well as registering new molecules or batches in bulk. You can register those molecules with or without structures, and it's basically the same process as we're going to go through now when you're adding this protocol data. So I've already registered my compounds, and I can associate the data to those compounds using their molecule names or synonyms. And this is the file that I've imported, and these are the fields in the vault. And I'm basically going to match them up. So I'm going to match a column to a field and click Apply. Once it turns green, that means it's mapped, and that column will be imported into the system. So we're going to do the same for all of these columns of data. I can create a new experiment and then click Apply. And then I'm basically just going to go through each of these. So I'm going to do PIC99. We have our ligand deficiency that was calculated outside of the vault, though you could calculate a ligand deficiency or a lipophilic ligand efficiency using one of those calculated readouts. So I'm going to import that data and then click apply. Once I'm done, I'm going to process the file, and this takes me to the third step of the wizard, which is a QC step. This will tell me what data is going to be uploaded, if there were any errors with my file. And so now this is telling me that the data is ready for review. We have 86 records that are going to be imported associated with the molecules that are registered, so I can choose to commit that data into my vault. So it's pretty straightforward. It takes maybe 2 or 3 times of importing data, and you pretty much get the hang of it. And it's the same really for any type of data set that you're going to import. Once that data is imported, I can now search for that data in my activity and registration database. So this Explore Data tab, I think, is where I had spent most of my time during my PhD, because I was searching and mining the data that had already been uploaded into my vault. So, I can click this green search button, and it's going to pull up all of the compounds associated with this HTS project that I have selected on the left. So these are the compounds that were registered. I can then look at the data associated with these compounds by customizing my report and basically turning on the fields that I want to look at. When you register small molecule compounds with their chemical structures, this is the list of calculated chemical properties that are actually calculated within CDD Vault by default using the ChemAxon cartridge. So these are, standard when you register small molecule compounds. If I want to look at that data that I just imported, I can turn on my HTS campaign protocol and then update my report, and I can see that associated data that I imported. And it'll turn on all of those columns of data. You'll see that as I scroll through. These are the chemical properties. As I get to the right-hand side, I have my, T-Brucei activity data, as well as all of the other data that I had imported previously. Now, some of these compounds don't have data for, the different strains of species. There's missing data here, and so I'd be scrolling through all of the compounds to look at the SAR for those compounds, which can be tedious. But to make my searching a little more productive, I can actually add some parameters into a search query at the top here to narrow down my search results. So if I click this search button, I can refresh my page, and then I want to look at that HTS campaign protocol, maybe for my T-Brucei inhibition with a PEC50 that's greater than 6, let's say. I also want to look in this other protocol that I have created for some ADME data. Maybe I want compounds with a kinetic solubility that's greater than 10 micromole, let's say. So I can also add a substructure search. So I can search by substructure in my database because these are fully rendered compounds. So I can draw in my structure. I can also copy and paste a SMILE string or mole text into this structure editor as well, and use this structure for my substructure search. I can add in some chemical property filters. Maybe I want compounds with less than 500 grams per mole. Let's say a CNS MPO score that's greater than 4. So this is a composite score used to determine whether compounds will cross the blood-brain barrier, which is essential for treating African sleeping sickness because those T-Brucei parasites do get into the brain. So the compound will also have to get into the brain in order to clear the parasites from the brain. So now when I click search, it will pull up all of the compounds that fit all of the criteria that I've specified in my search query in a report table at the bottom. So there are 28 results now, whereas before we had 78 results. And again, anytime I generate a report table like this, I can customize my report and I can turn on or turn off any data that I want to see. Maybe I want to see my Hep G2 to PTC50 and update my report. Once I have compounds in a report table, I have the option to export all of this data to an Excel file or a CSV, even an SDF to include the structured data as well as a zip folder if you have files attached into these fields that are stored in the report table. I can save these compounds to a collection. Maybe this is the criteria that I think, deem a compound to be a hit. So I can add this collection now to my project, and then the other members of the project can view this collection at any time. And then I can share this collection with my colleagues. If I had other members in my vault, I would be able to send an email with a link to this collection saying, hey, these are really good hit compounds. We should take a further look at them. Collections are static lists of molecules. So if I wanted to update that collection, I would have to rerun a search and then save it, with the same name as that already existing collection. But I can also save my search, which is a little more flexible, and it will save all of the parameters of my search query, as well as the layout of my search report table. So then, a week later, I can go back and I can run the same search. And if any new compounds or new data has been added to the database that fit the criteria of my search, it'll then be pulled into that search report table. All right, so these are some data points that, I would be going through to basically narrow down which compound I wanted to work on for my PhD project. So using this data, I wanted to narrow it down. And scrolling through Excel files or tables like this, as I said earlier, can be a little tedious. So a great way that I liked to narrow down my search results is through our visualization module. So I'm just going to pull up all of the compounds again in this project, and I'm going to turn on all of the data I have available. And then I'm going to update my report. So now I have many columns of numerical data turned on, and I can take these 78 compounds and all of this data and launch it into our visualization module. So this opens up in a new tab. By default, you get a scatter plot at the top with a duplication of that search report table down at the bottom, and then some filters over on the right-hand side. I can customize my axes. So typically what I would do is look at my T-Brucei either PEC50 or PIC99, right? So if we want the compounds to be fast acting, we want a PIC99 value that's greater than 6. This is basically, or that determines cytality. Sorry, I misspoke. So we want compounds that are killing parasites instead of just inhibiting growth. So a higher PIC9 value indicates that the compounds are cytal. I can then change my y-axis as well. So I'm going to change my y-axis to my lipophilic ligand deficiency. So this tells me basically the quality of my compounds and whether or not they're making favorable interactions with the targets within the parasites. I can now filter my compounds by, let's say I want all of my compounds to have a log P less than 3. So I can add a filter here, and you'll see that as I select compounds in the filter, it'll update my scatter plot on the right-hand side. But now I also want to look at my CNS MPO score because I want compounds that enter the brain. So I'm going to filter by that MPO score. I can also change the color and the size of the points in the scatter plot to be mapped to a different parameter. So now I'm actually going to map the color to that kinetic solubility because we want compounds that are water soluble, that they can easily get into the body. So once I've done that, I click done, and now I can have a 4-parameter scatter plot. I didn't map the size to anything just because I like my points to be the same size. So if there's an area of interest, so if I want that PIC 99 value greater than 6, I can select an area of interest. I want to get that. Oops, I missed that other point up there. So I want to get that other point. And I can zoom in on an area of interest. Now, based on all of my criteria, I've pretty quickly narrowed down my molecules to a list of 10 that are displayed here. And the 2 that are green are the compounds that are soluble. And so I can hover over those points and see which structures they are. If I hold down my shift key and then click, I can actually paste the structure into the plot area. Move it out of the way, shift click. And then I can, turn on other parameters. So maybe I want to also display that, TBB PEC50, the lipophilic ligand deficiency, and it will update my compounds there. So I pretty quickly narrowed down my compounds, and based on the narrowing down of these compounds, I had actually chosen this molecule that you see here, C57, as the compound that I wanted to progress further as part of my PhD work. So this was my hit compound, and basically all of my work was then focused around lead optimization for this molecule. So this molecule is also known as NEU 1200. That's what I remember it best by, so that's what I'm going to refer to it as. It's an azaindole. You have the pyrazole down at the bottom, and this substituted benzene on the other side. So where I started with my work was basically to resynthesize NEU 1200 from scratch, basically so we could retest it and validate it that it was in fact a hit molecule in another T-Brucei whole cell assay. So I used the ELN to set up my chemical syntheses. Since, as I said earlier, the ELN is just a blank page when you create a new entry, I can really add all different types of data. So each of these links is actually a link to another ELN entry with the other steps of my reactions. So I basically started with step 1, and from step 1, I duplicated my entries to end up here at step 6, which is my final product, or NEU 1200. I used the insert structure reaction, the little benzene icon, to draw in my chemical reaction for this last step. It'll recognize molecules on either side of the arrow as well as above and below the arrow. So when I draw in a reaction, it'll automatically generate a stoichiometry table. At first, it only had the 2 compounds that I had drawn in my reaction, but I can actually add a reagent in manually by clicking this add reagent or add solvent button. And then I fill in the appropriate information about the other reagents that I used. in this reductive amination. As you can see here, I can edit all of the amounts used in my chemical reaction, specify my limiting reagent, and I can even register my intermediates as well as the reagents that I used into my CDD Vault. So a lot of customers actually even have a separate vault for their chemical reagents, where they can keep track of the inventory of their reagents as well. And then we do have a feature where you can link across vaults and then pull in the information from other vaults here into the stoichiometry table. Once you finish the reaction and put in your mass, it'll calculate a % yield based on the purity of your final product. And then I have the option to register a molecule here from the ELN page as well. So if I draw a structure that is not registered in my database, you'll see that the molecule allows me to register it. If I do draw a molecule that is already registered, it'll display that molecule name automatically. So this is recognizing NEU 1200 as that molecule C57 in my database that's registered. I can insert links to my activity and registration database, as well as if I click this insert link button, the first suggestions that pop up are actually the reagents from my stoichiometry table. So I've added these links into my procedure now. And if I update the amount that I use in my procedure, it'll also update the amount in the link that I pasted below. So I'm not having to write out the whole IUPAC name as well as keep track of amounts. Basically, I can update it in one place and it will update it for the rest. You can see I set up different sections for the rest of my procedure. I did my analysis and purification and attached PDFs of my LCMS and NMR. And then down at the bottom, kept track of where the compound was being shipped to and where it was stored, and any other information. So now from here, I can register this compound. So I have other batches of this molecule. So what CDD Vault does is it checks your entire vault for duplicates of your parent molecule. If a molecule had never been registered before, you would get a new molecule created or new entity if you're not storing small molecules. And those molecules are assigned molecule names that consist of a prefix that you can configure and choose whatever you'd like. Here I just chose C. In our other vault, our prefix was NEU, and that's why I knew this compound as NEU 1200. And then that molecule name is then followed by the next available sequential number in the vault. Now, since I'm registering a molecule that has already been registered previously in this vault, I'll get a new batch created of this molecule. I can capture any information about salts or solvents present. Maybe I took this off the PrEP HPLC and this is actually the TFA salt. So I can specify that this is, one equivalent of TFA. And then I can fill in any information about the compound that I want to register. These fields that you see here are also configurable by the users or by you as a user of the software. So if there is other information, I can actually create a field and capture that information. So these fields are what we call batch and molecule fields, and they can store data that are text, they can be numbers. You can also create pick lists to control the vocabulary, as well as upload files and attach files to these fields as well. And then finally, you specify which project this molecule belongs to or this batch belongs to, and then you can add that new batch from the ELN. As I scroll through this batches tab, you'll see that the various batches of this molecule have been registered from these specific ELN entries. So it automatically creates this breadcrumb link back to that entry where the compound was registered from. So if I go back to that, if I click that link, it'll take me back to that registration page. I can add links here in the ELN as well to my database. So if I wanted to link to a protocol of data, I can add a link in that way. This is my actual protocol. If I wanted to insert a link to a run of data, let's say, what is it, 2021? 07? Here is that run or that experiment of data that I imported. And these are all clickable links. So I can open this up and it'll show me that data that was imported. And then, any of the fields that you create, any of the protocol readout definitions that are created are all searchable. So, I can search by the inventory current amount for my compounds, as well as any of those fields that are set up. So, I showed you basically a couple months worth of work all in a couple of minutes. And, the system really helped me to narrow down the compounds that I wanted to work on, as well as streamline the process. And then it's all here, on the cloud for our collaborators to view, as well as my PI who wants to keep track of me in my ELN. You can look at that information that's up to date. As changes are made to the ELN, it auto saves. And then we also have the features to finalize as well as export an audit trail as well as duplication. So, hopefully I've showed you, most of CDD Vault and the capabilities that it has. Obviously there are a lot more, but I don't want to talk for hours. So, I'd like to take this time to open it up to any questions you might have about the platform, neglected diseases, and anything like that. So with that, thanks for your attention, and I'll be happy to answer any questions.